Hello friends. Today I'm going to discuss about your elongation and termination steps in translation. So I have already discussed about this initiation steps of translation, and if you want to go through those to know about the initiation, how it takes place, you can go through this link I have given here, and you will come to know about it. So already I discussed that in protein synthesis, three steps are there: initiation, elongation, and termination. So now today I am going to discuss about elongation and termination. So how does elongation in translation take place? So after the formation of this 70S initiation complex, which was discussed in the initiation steps, what happens? The elongation take place. And then the elongation, there are three steps: there is binding of amino acid tRNA, peptide bond formation, and translocations. So here is a general view, general picture which I have shown you, which is applicable for both your eukaryotes and prokaryotes. So here in the picture you can see that after this methionine tRNA has come to the position of P, and it has base paired with your initiation codon AUG. The A site is now empty. Now it is ready to take new amino acid tRNA. So now this new amino acid tRNA, with the help of your GTP, which get hydrolyzed, and in the presence of elongation factor, it comes and get positioned itself into the A site. So whatever here in the A site, it position itself only when it makes your that anticodon of tRNA makes base pair or complementary base pair with the codon of the messenger RNA. Only then it gets stable there, and this way your this new amino acid tRNA get position itself. And after that, in the presence of your enzyme, that is peptidyl transferase, peptide bonds are your formed, and these peptide bonds are formed after one after another. New amino acid transfer RNA comes with these new with the assist of your new codons will come in the mRNA, and it will move in the direction in this way, and then your more and more codon will come. This tRNA will base pair with this codon, and then this transfer peptide bond will be forming. So this is called a translocation. So in prokaryotes, the same thing happens, only the elongation factor T is applied, and in association with GTP, this binding of amino acid tRNA at the A site takes place. So first, the elongation cycle begins with insertion of amino acid tRNA into the empty site on the ribosome, which I have discussed. Second, if the codon on mRNA matches with the anticodon of tRNA, then GTP is hydrolyzed, thereby you are positioning the amino acid tRNA in the inside, which I have told you already. And then elongation factor transfer this amino acid tRNA A site by binding to it. Now in the next step, peptide bond formation and translocation. So what happened that when you can see in this figure that this tRNA has brought valine, and it had made a complementary base pair with the mRNA, and then this valine will go and get added here, forming a peptide bond, and thereby this new amino acid tRNA containing new this is the codon, and it is bringing the information which is anticodon will match with this codon, and again this is for ASN. So for this it will go and base pair with it, and this way until and unless the stop codon or termination codon don't comes, the polypeptide chain will keep on growing. We have seen it is keep on growing, and here this tRNA it get released for attaching new amino acid to it. So in prokaryotes, we should remember that it's a very simple process. Translocation requires a third EFG, that is translocase, causing hydrolysis of GTP, whereas in eukaryotes it requires EF2 and GTP. So GTP get hydrolyzed and supplies energy to move mRNA. So steps involved are already have discussed. That is, this prokaryotes, formalin methylene tRNA will be there, whereas in eukaryote it is not formalin, which is uncharged. It leaves the P side. Then second tRNA, it bound dipeptide moves to the P side. Then mRNA moves to distance 3 nucleotide towards the 3' end. And after translocation, the A site is opened up to accept the incoming amino acid tRNA, which matches to the next codon of the mRNA. And this elongation factor TU, in case of prokaryote, delivers the next amino acid tRNA to the empty A site. So here I have listed the elongation factors which are required for prokaryote and eukaryote, and with the general functions. So functions I have already discussed. Only the difference is that in eukaryotes your E prefix is used, and also instead of EF, TU, TS, it is alpha, beta and delta. And translocation factor, it is in prokaryote EFG, whereas in eukaryote it is EF2 for ribosome translocation. Now this comes after peptide bond formation and growing polypeptide chain is now growing. The next step is termination in translation. Translation has to stop at a point. So basically we need a stop codon and release factor for that. Stop codon like UAA, UAG and UGA are the 3 stop codon which don't have any complementary base pairing or active codon in the tRNA, we can say. And that's why when this codon comes in the mRNA, tRNA stops coming, and then your translation also stops. So here you can see if you stop codon, so they do not have specific tRNA. And then stop codon, when it occupies your A site, this is your A site, and release factor, RF factor, it recognizes it. Then RF here and GTP complex is formed, which in association with peptidyl transferase clips the peptide bond in the A site. It will clips it in the A site. Then water molecule is added instead of amino acid. So this hydrolysis releases protein and tRNA from the B side. So water molecule is added, and instead of amino acid, because now no polypeptide chain has to be made further. And this hydrolysis releases protein and tRNA from P site, and ribosome dissociates into 2 subunits, and mRNA is also released. The polypeptide chain is also released, and thereby termination takes place. So here also in prokaryotes, terminations release factor RF1 and RF2 are involved in this termination codon recognition. And what is the role of RF3? It binds to GTP and stimulates the binding of RF1 and 2 with the ribosome. Second, you can see termination in prokaryotes. It is also in the same way that RF binds to stop codon and goes to the polypeptide chain from A to the B side. This RF factor will come and it will bind to the termination codon or your stop codon. Okay, so UAG, and the extra bond between the polypeptide chain and last chain is then hydrolyzed. So this binding of RF to the terminating codon causes water to act as the acceptor of growing peptide, not new amino acid is added. Same in the prokaryotes also, the same state which I have discussed. So now the release of polypeptide chain is followed by dissociation of mRNA and tRNA, and dissociation of 30S and 50S. Now EF3 will come, and now associate 37 to prevent reassembly in the absence of mRNF material. So elongation and termination step is very simple steps. So in eukaryote, termination is carried by a single release factor that you should remember, like ERF, and in prokaryote RF1 and RF2 was used. So finally this long factor like IF3 in prokaryotes prevents reassociation of ribosomes when synthesis is completed. So lots to learn, keep learning. Thank you, and subscribe my channel if you like it.